So I'm very happy now to welcome Dave Weitz, who is joining online from US. I think everybody knows him, but I will still go through a bit of his works and what he's been doing. So he's a professor of physics and applied physics at Harvard University. A director of Harvard's Material Research Science and Engineering Center, co-director of the BASF Advanced Research Initiative, and a member of the National Academies of Science and the American Academy of Arts and Science. His research are focusing on soft matter physics, biophysics, and biotechnology. And he's also the co-founder of several companies. I can cite, for example, Capsum or Hi-Fi Bio, which are close to our ecosystem. So Dave, thank you very much for being with us, and the floor is yours.
Okay, thank you. You can see me now. Let me share my screen. Okay, I think I can see my screen in the background. Well, thanks very much. Congratulations to Fluigent. I can't believe it's 15 years. Maybe you can't as well, but congratulations. And it's a pleasure to be here. I have to say, I wish I could be with you, and the pandemic is not what's preventing me from coming. It's that I'm very busy teaching this term. Otherwise, I would definitely join you in Paris. And congratulations to everybody who's there. So let's see, I maybe say a couple of words about our involvement with Fluigent, just briefly. We were very lucky to have some interactions with the local people here. They've been very, very helpful to us. They put a couple of the pumps, the Fluigent pumps in our lab, and now everybody wants to use them. Moreover, I think that a lot of people who've left the lab and started companies have turned to Fluigent for their pumps. And so again, I congratulate you on all the excellent work that you've done and that you continue to do. Today, I'm not going to talk about any of our startup companies. I want to just talk about some of the work that we do in my lab, mainly thinking about new techniques, new ways of making measurements of fluids, both for microfluidics and somewhat fluids at the microscale. And the work has been done, let's see if I can find my cursor, laser pointer. The work has been done by, a lot of what I'll tell you about was done by Stefano Amé, who was a postdoc with us and now is actually in Paris, probably right near you at the SPCI. There are several students who've contributed to the work. I'll tell you quite a bit about what Anchi Chen has done. And we have some collaborations both from people in Australia and in China. So I'm going to tell you sort of a potpourri of different things. I'll tell you two different techniques. I'd like to introduce them, explain a little bit about them. The first, I don't want to tell you too much about why we do this. I want to just show you what we do, but let me give you briefly an introduction. This was a discovery that was made actually from a former student from our group who was working in Dresden at the time, about 10 years ago, Cliff Brangwyn. Discovered that in cells, you often see a phase separation process where proteins can undergo what he called the liquid-liquid phase separation, but essentially you get these liquid-like drops of proteins, of concentrated proteins. I would say they're condensates, that's what I'm going to call them, in the cells under certain conditions. And this has a lot of important implications for biology. We even have a little startup company that's trying to exploit them, but that's not really what I want to discuss today. What's interesting about them is that you can reconstitute these small drops of concentrated protein. So think of them as a phase separation where you get a concentrated region of protein in a sea of unconcentrated protein. and they form a small drop. And we wanted to learn, do something to measure their properties. And so we did a technique that's actually fairly old. It's called micropipette aspiration, but I don't know that it's ever been applied to something as delicate as these. And we were able to do this only because we had a constant pressure pump that we have from Fluigent. And this has sort of inspired a lot of people in our group to try and look at this behavior and use micropipette aspiration. So here is a condensate. You can see this is, it's reconstituted in vitro. It's about 20 microns in diameter. This is a pipette, and this is operated, it's controlled, the pressure is controlled by just a Fluigent constant pressure pump. And if we suck on the condensate, it looks something like this. And so if we think about this, here's the control, this is the pressure, and we have a pressure here that we set with the controlled pressure pump. We can measure a radius curvature here, we can measure a volume here, and we have a pressure difference that we know. And so we know the pressure difference, and then we can watch the way the drop behaves. So once you achieve above a certain critical pressure, it just flows right in. That's what you saw. And from the way it flows, actually from the shape it has, you can measure the surface tension. And from the rate of flow, you can measure the viscosity. The interesting feature about this is that the surface tensions are extremely low. And so again, this is just showing what it looks like. If I do several of these aspirations, now using different pressures, you can see what happens. Here is a fairly high pressure, it goes in pretty quickly. A lower pressure, it goes more slowly. An even lower pressure, it goes even more slowly. You can measure the rate, you can measure the shape, you can plot the different parameters. And from this, we can make some measurements of the structure of these things. We find that the surface tension is extremely small. It's about 10 micronewtons per meter. So it's maybe a factor of 100 to 1,000 smaller than a typical water drop, a very, very small surface tension. And the viscosity is actually fairly high. It's about 1,000 times water. So it's very viscous, but a very, very low surface tension. Now, I won't go through the details about this, but if you expose these things to UV, then you find that they gel. If they gel, they form the same. The drop looks exactly the same, but now it's a solid. Now you have to ask different properties. If you pull on or you put a pressure on these things, and you can increase the pressure, you can see that here the pressure is increasing as a function of time. Let's see if I can play that again. Let me just go like this so I can play it again. You can see it goes in, then we hold the pressure constant and then drop the pressure, and it springs back. So it's clearly elastic. You're putting pressures of order 2 kilopascals, and from this you can calculate an elastic modulus, a Young's modulus of order a kilopascal. So you can have these very weak, very viscous, but very low surface tension drops. That's just a phase separation of proteins. These particular proteins are fused sarcoma proteins. They're associated with a certain disease, and they're phase separated through the addition of both salt and some crowding agent, basically a depletant polymer. And by exposing them to UV, you can turn them into a gel. Okay, what I wanted really to do was to show how easy these micropipette aspiration measurements are, yet how valuable they are, how informative they are. And again, we just couldn't do them without having a constant pressure pump, which makes it so easy to do that now many people in our group are beginning to do these kinds of measurements. Now I want to change gears a bit, talk about a different measurement. This is something that Stefano invented when he was a postdoc with us. And he asked a certain question. He said he would like to measure very, very small motions, but in a very large field of view. And he made this little cartoon. He would like to measure this ladybug walking on a leaf far away in the mountain, but have a large field of view. And how can you do this? The problem is that if you go close and you look at it, you can see the ladybug, you can see it move, But as you go farther and farther away, you lose that resolution. And the reason for that is that you're always trying, if you want to see the motion of the ladybug, you're always trying to resolve an object, resolve the motion of something like whatever it is. You have to resolve something and then see how it moves. And that gets very difficult when you're looking at very high resolution, or when you're looking far away, you want to get a large field of view. So if you have a large field of view, you lose the resolution. So how can you break this dilemma, this contrast? And what Stefano realizes the way to do that, of course, is to take advantage of interference of light. So if you have two particles and they are exposed to a laser beam, the light that's scattered from them interferes with it, the light from each particle interferes with the other particle. And so if they move, you get this interference pattern, and the intensity then fluctuates according to the way the motion of the particles. From looking at these fluctuations, you can do essentially a correlation, a time correlation function. This is standard dynamic light scattering, but you don't have to do necessarily dynamic light scattering. All you want to do is look at a speckle, look at an interfering pattern, and measure it the way it is correlated. And so Stefano realized that you don't need to just look at a small volume. You can image a large region, but you image now not the light itself, but the speckle, the scattered speckle of the light. So here's an image where you can see the speckles here. This is an image of a channel in a microfluidic device. The fluid is flowing in this direction. He seeded the fluid with some small particles just to get a little extra scattering. And you can see there's speckles from the sides which are stationary, speckles from the fluid which are moving because the fluid is moving. This is what it actually looks like. If you take an image of the camera, actually what he's done here, sorry, he also he's illuminated it with two laser beams, taken it with a single camera, and by doing that, he can look at motion in two directions, two wave vectors, which in this case are perpendicular to one another, so you can see motion in the X and the Y direction. And these are the speckles that are fluctuating in time. These are the speckles that are constant in time because they're due to the walls that are not moving. And what Stefano does is just looks at the correlation of each of the speckles, takes a little region around it to get a little bit of an average, and he can calculate that. The reason he can calculate that is he knows the flow of the fluid. They're flowing in this direction. So you have to imagine, just go back and think about the speckles. They're being created by flow of particles. You're looking at the relative motion of the particles, but you're doing an imaging experiment. So you're imaging across the whole sample. And okay, you can calculate everything, and you can actually calculate the whole correlation function. It looks like this. The yellow is the calculated. The solid points are actually measured data for different velocities of fluid. You notice that you get this decay, you get some oscillations. You get oscillations because you have the relative motion of the particles. You get some of them moving faster than the others as they go through the center. It's a Poisson flow. So you can do this. In fact, you can do this for all different kinds of geometries. I won't go through the details of this, but this turns out to be a very powerful method because not only can you look at the relative motion, but you can actually see when the fluid is flowing really fast, you can see the speckles just whiz by and flow through the laser beam. And that's a different type of analysis. You still can do the analysis, and you still can get some valuable information about the profile of the velocity. Similarly, if it's going very slowly, then the dominant motion is the diffusion of the particles, and that too can show up. And you can see this. I won't go through the details, but you can analyze each of these correlation functions. These are just correlation functions for different velocities taken with different scattering geometries to exploit all of that information. However, this becomes even more powerful if you look at something that's more elastic and you want to see a large field with tiny motions, as you would see in some strain-related measurements of an elastic medium. So here is a small microfluidic channel that's filled with a colloidal gel, and Stefano injects some water in it to create a crack in that gel. This is what it looks like. And then he does exactly the same thing. He sets it up so you have, now you're imaging the whole sample here. The gel doesn't scatter that much, so you can make an image of the speckle pattern shown here. And this is what it looks like. The water is being injected here, and I'll just play this movie. Okay, where is it coming us? Okay, here we go. Here you can see the speckles, and they're moving downstream as the crack goes. This is what the speckles themselves look like. They don't completely decay, so you don't have to actually look for a stationary pattern. You don't have to have something that repeats itself in time. You can see something, this movie is being played over and over again. You can see something that's just moving slightly in time, but you still can calculate by averaging over the region, averaging a few speckles, you can still calculate the correlation function. And from this, you can actually now calculate minute displacements. These are sub-wavelength displacements of order a few nanometers, and you can add them up and watch them displace in time, and you can look at the difference between 2 images and 4 images and 8 images and see the added displacement. Sometimes they displace a large amount, sometimes it takes many times to go. So you're basically measuring the speed, the velocity of the displacement, and now you can plot this. Instead of plotting the actual displacement, you plot the magnitude, the speed of the decay, and you can see as the crack propagates, it moves here, and these are the large, fast oscillations. You can do again the same thing by adding a second laser. So now you can get a whole displacement profile, and you can see just what you expect. The crack is tearing it apart. You're getting a whole strain field as you measure the sample as you displace it. And here is just doing the same thing, just looking again at the amplitudes of this, and you can see this, a very, very powerful technique to get the whole strain field. In fact, you can go in and look in great detail and watch the whole crack develop. So here's just a imaging, because you can look at this in all dimensions, you can image this and you can see the full dimension of the crack. And this is just the crack moving apart. This is the strain field that's moving apart. The crack is moving in this direction. The water is being injected in this direction. And you can see that the gel is being pulled apart on either side. And you can see the whole strain field. So now you're measuring strains, small linear motions of sub-wavelength. The smallest amplitude is about 5 or 10 nanometers to some number of microns, because you can do this and you can do it over and over and over again, so you can measure the full displacement. You get the full displacement field. You're not limited just to the 2 dimensions. You can also do 3 dimensions. It gets a little more complicated. Now what you have to do is you have to add 2 more scattering vectors. The reason you do that is you do small angle scattering. And so you have a scattering in this direction and a scattering in this direction. You have, by using a back scattering and forward scattering, you can take the large large component of the scattering vector and subtract it, and you're left with 2 that just add. The 2 then add in the 3rd component. That's the up-down component. So by doing this, now you have to do actually 4 detections. You can get a 3-dimensional view of the image. Here's an example of this. This is the crack that's propagating from the beginning, and this is the full 3-dimensional image. It's X and Y is the arrows, and the color is the height, or the sort of the color map gives you some sense of the height. Here it is in different times, locations, but you can get the full 3-dimensional image of something that's scattering with very, very small displacements, but very large field of view. So we call this dynamic speckle holography because we're looking at the dynamics, we're looking at the speckles, and we're really doing holography because we're doing it in 3 dimensions. Okay, with that, I hope I've not taken too much time. I wanted to keep it short so we have time for a few questions. I've tried to tell you how really the great luck we've had of using the Fluigent controlled pressure pumps in such a simple way to do micropipette aspiration has really inspired a lot of different work in the lab. I wanted to show you one really cool example. I think one that we're very excited about. And I wanted to show you a second measurement technique. It's not so complicated. It can be set up fairly easily. It was invented by Stefano, and we are using it for a lot of things, but mainly visualizing strain fields in 3 dimensions. Very, very small displacements over a very large field of view. So with that, let me thank you for your attention, and I hope I can leave some time to answer some questions.
Okay, sorry, you didn't hear me. So I was just thanking you for the great talk you just gave us, and asking if there are any questions here or online.
Can you hear me?
Can you hear?
Yeah, I can hear.
Super. Thanks, Dave. This is Charles Beirut. I'm curious how you actually...
Charles, nice to see you.
Yes, likewise. I'm curious how, if you can tell us how you actually make the lipid droplets, and are they actually phase separated the way that they would be in biological systems? And if you have some time, tell us a little bit what's inside them. Why do they gel?
Okay, I can tell you, first of all, they're not lipid droplets. That's the very cool thing. These have no membranes. That's what's really cool about them. They're just a phase separation of this particular protein in the solution. It undergoes phase separation, just like any normal phase separation. So it phase separates into a protein-rich region and a protein-poor region. The protein-poor region is everything around it. The protein-rich regions are these spheres. They're spherical because there is a surface tension. It's very low, but there is a surface tension. But the remarkable thing is that there are no lipids, so there are no membranes around these things. And for FUS, the way we do that, I think, is we control both the salt concentration and the PEO or PEG concentration. So it's just a mixture. And it's a good question to know whether these are exactly the same as they are in cells. This protein will phase separate in cells without the presence of lipids, just because of the occurrence of the protein. But of course, the interior of a cell is a much more complex environment. So is it true that the phase separation is driven in as simple a way as it is in vitro? I can't tell you that. But that's one thing that we and many, many other people are looking at to ask whether there is that kind of correlation or not. And then you asked how I gel them. And we honestly don't know why these particular condensates gel. We noticed it when we were studying them with a confocal microscope, and we were using a fairly high excitation energy, a short wavelength laser, and we noticed that the ones we were studying looked very different than the ones that were neighboring but had not been illuminated by the laser. We've since figured out that if you expose them to UV, they seem to gel. Why that happens, we don't know yet. More generically, in cells, the protein condensates tend also to sequester RNA, and RNA is a good gelling agent, again, usually if you expose it to UV. So it can occur in the cell. It turns out there are many other reasons why it could gel, and it often does seem to gel when they form in the cell. And the correlation between the in vitro condensates and what goes on in the cell is not clear yet. But the beauty of studying these is that you can see some correlation between the in vitro and the in vivo. So you can make a lot of interesting measurements with these in vitro systems.
Thank you. There is one online question. Do you think you can measure the elasticity of these hydrogels using these micropipette aspirations?
And the answer is absolutely. I showed you one example where you do, and you might ask, could we measure the frequency dependence, and can we measure it really precisely? And the answer is, I think so, but then that depends very much on the interpretation of the shape and the dynamics of it. But certainly you get a sense of that just because you could see that I could apply a pressure of 2 kilopascals and it wouldn't it would not completely deform the structure. It deformed it roughly 10%. So, you can figure that the elastic modulus is of the order of a few kilopascals. And that's when we did it more precisely, that's what we measured. But yes, I think you can. And that's the beauty of this technique, is you can measure all of the properties using just this beautiful and really very simple technique that really becomes really trivial to do with the Fluigent pumps.
Thank you for your kind words. And maybe the last.
Really, it's true.
Thank you. Maybe just one last one. Can the method for studying the movement of small particles in large FOV used in centrifugal microfluidics?
That's an interesting question. So I would say I think so. I think because what we're doing is taking snapshots, and so you'd have to take a couple of snapshots. You might be able to even imagine, if it's not too slow, if the movement depends on the scale of the motion, you could imagine just taking snapshots of image on each rotation and comparing the speckle fields. It would be not trivial, but I think doable. But my best answer is that the real whiz, the person who knows how to do this better than anybody else in the world, is the inventor, Stefano, and he's right there in Paris. You could ask him.
Excellent. So thank you very much again, Dave. Thank you to Séverine, Raphaël, and Charles for the great speech that you gave today, and for your time and availability to be part of the celebration.